Our next speaker is Dr. Jeff Keil from StemCell Technologies. Jeff used to be our field scientist covering the region, so nice to have you back, Jeff. His title is Modeling Neural Inflammation in a Dish with hPSC Derived Cerebral Organoids and Microglia.
Thank you, Shasha. Can everyone hear me?
Yes, we can.
Oh, perfect. So yeah, I'd like to thank the GESC for organizing this event and also for allowing us the opportunity to speak. Today, I'll be talking about building a better in vitro neuroscience model. So just a little bit of a background here. So my name is Jeff. I am now the Strategic Alliance Manager for our cell culture portfolio at StemCell Technologies. It's a fluffy title, and really what that means is that I get an opportunity to chat with groups like yours that are at the forefront of research and often clinical translation. So today I'll be talking about some of our efforts towards creating better in vitro models for neuroscience research. And while I will spend a little bit of time detailing some of our current and upcoming neural products, I'll be doing so from kind of a big picture point of view. I'll keep the product spiels to a minimum, I promise. But do know that if there are any particular reagents or kits that you're especially interested in learning more about, we're always happy to schedule a meeting to do a deeper dive. I'll spend a lot of time today talking about sort of our approach to neural product development, co-culture, and modeling neuroinflammation. So just for those who are not familiar with StemCell, we're a biotech company located in Vancouver, British Columbia. We offer over 2,500 products that fully support all aspects of life science research. We have both an immunology and a cell culture side, and we offer products from really every step of the workflow, from cell sourcing all the way to cell culture and analysis. We also provide contract assay services and training and education workshops, and also have a really fantastic team for translating your discoveries to the clinic. So today I will be focusing mostly on our neural media portfolio. And this portfolio is rooted in our desire to constantly innovate with the ultimate goal of creating a better in vitro model. So StemCell was one of the early adopters of hPSC technology, and we've put a lot of effort into developing reagents really for every stage of the classic hPSC workflow. Whether you're generating iPSCs, maintaining, banking, editing, or differentiating, we have reagents really for every step. And we've really put a lot of time and effort into offering a wide variety of differentiation kits, which span over 40 different tissues across the 3 major germ layers. So I'm not going to go through each one of these in detail just because it'll take too much time, but I'm mainly showing this to give you a sense of the effort and commitment we've put towards hPSC technologies, both in terms of mesodermal and endodermal, and also ectodermal and neural tissues. And of the 3 major lineages, our neural ectodermal products are by far the most developed, with over a dozen 2D and 3D organoid-based kits. Now, in terms of modeling neuroinflammation, we have many of the relevant glial cell types, including astrocytes and microglia, as well as a variety of neural cell types that would interact with them normally in vivo. So when we're developing our kits, we put stock into a few key factors. And we're always doing this with the idea of creating better in vitro models. And there's certain characteristics that we really kind of value. And the first has to do with sort of physiological relevance. So a lot of our kits are developed with a faithfulness to the developmental biology. We're always keeping in mind these kits support physiological levels of activity and function, and that the relevant cell types, or when possible, the relevant cytoarchitecture is there. We also want these to be easy to use. I think this almost goes without saying, but we don't want the complexity of our kits getting in the way of your experimental results. we want to make sure that you don't have to have extensive PSC experience to use them. And we also want to make sure that there's no cumbersome steps or specialized equipment that's usually needed. And we also, I think, almost more than anything, want to make sure that these kits are consistent and flexible. Pluripotent stem cells are inherently variable, and we spend a lot of time up front making sure that these perform consistently across lines. And we also know that your time is very valuable. And I'm sure you don't want to be stuck in the lab for 80 straight days changing media on a differentiation protocol. So to this end, we do try to develop as many pause points throughout these protocols as possible, making sure that you can freeze down at various progenitor and precursor stages with minimal loss to efficacy and viability. So one of the things that I want to focus on a little bit more is the physiological relevance. And there are a number of approaches for deriving a neuron. At StemCell, we've tended towards a classic stepwise directed differentiation, as this mirrors neural development in terms of patterning and timing. And there are certainly faster approaches. I think the transcription factor based approaches come to mind. And while those approaches are actually fantastic, especially in terms of speed and purity and ease of use, they are often lacking when it comes to certain types of function. For those that are familiar with the NGN2 based methods of deriving neurons, one of the things that was originally sort of found in those first couple of papers is that in the absence of an astrocyte feeder layer, the neurons didn't efficiently synapse. And thus they didn't really, fire action potentials at a normal level. So there is clearly something that's kind of lost when you skip certain developmental stages within a differentiation protocol. So currently at StemCell, all of our kits use this classic stepwise approach. And these are just kind of examples of how we start with PSCs, generate neural progenitors, and from there we'll generate the precursors of the different neuronal populations. And another way we consider the physiological relevance of a neural model has to do with activity. So, which for neuroscientists out there, I hope this rings true, but as activity underlies all neural function and dysfunction, and thus we strongly believe that an ideal culture system is one where the physiological levels of activity are supported. So with this in mind, we've incorporated this reagent called brain Fizz into a number of our differentiation kits and support protocol. And if you're not familiar with brain Fizz, that's all right. I'll do a quick run through here, but the story is kind of interesting. So basically, Cedric Barty and the Gage lab out of the Salk Institute found that traditional media did not really support physiological levels of activity due to the presence of a whole host of things, including neuroactive peptides and non-physiological levels of osmolarity and hyperglycemic levels of glucose. And so, what they did is they set about developing kind of a new media, aka brain Fizz, which in fact supports these higher levels of activity. And this in turn translates to improvements in maturation and function. And it's really been kind of widely adopted, in a relatively short period of time. And, it's one of these things that, StemCell really kind of jumped on the bandwagon. Now, as part of creating better in vitro models, we're also kind of concerned with incorporating relevant cell types and cytoarchitecture into our model considerations. So within this sort of continuum of modeling complex neural phenomena, you have a bunch of options. You have your pure 2D cultures, you have your, slightly more complex co-culture systems, and then you have your 3D organoids. And all of them have their advantages and disadvantages as a neural model. So your simple 2D cultures are clearly advantageous for sort of any higher throughput application, but their simplicity may be a bit of a drawback depending on what types of questions you're asking. So if you're interested in complex physiology or cell-to-cell interactions, a co-culture or 3D model might be a better choice. And those systems allow you to kind of capture developmentally related phenomena or interrogate the interplay of multiple cell types or regions in a way that's not really possible in a monoculture. So the trade-off is obviously sort of throughput and time. Generally speaking, your organoid and your co-culture approaches will generally take a bit more time and require some more specialized steps. So like I said, ultimately, it's going to come down to your research goals. So at StemCell, we are becoming more and more interested in these complex systems, especially co-culture model systems. So much so that we've actually developed multiple detailed co-culture protocols for both 2D and 3D applications. So here on the box on the left are just an example of some of the ones that we've developed in the last year or so for neuron and astrocyte and microglia bi and triculture, as well as microglia and organoid co-culture. We strongly believe that 3D organoid approaches and co-culture approaches are really going to become kind of the standard for how a lot of neuroscientists kind of model different aspects of neurophenomena moving forward. So this finally brings us to kind of neuroinflammation, and just as a bit of a reminder, neuroinflammation is the activation of the brain's innate immune system in response to an inflammatory challenge. And neuroinflammation is fairly ubiquitous. In a sense, neuroinflammation is a broad phenomenon playing a role in most aspects of neurobiology. And it's heavily implicated in all manner of neurological disorders. So one thing to note is that microglia and astrocytes are key regulators of this process. So it stands to reason that faithful modeling of neuroinflammation is going to require these critical cell types. Besides neurodegeneration, astrocytes and microglia also play a large role in maintaining the health and homeostasis in non-disease states. So it's definitely something to keep in the back of your mind, no matter what type of paradigm you're modeling. For the purposes of the rest of this talk, we'll be focusing on microglia and astrocytes in regard to neuroinflammation. However, there are other contributing cell types, including oligodendrocytes, macrophages, obviously neurons, as well as the blood-brain barrier. So the number of studies that have come out really in just the past few years utilizing these more complex co-culture approaches to model neuroinflammation are almost too numerous to count. And this isn't just conceptually a nice system to employ. There are really clear functional benefits to a lot of these approaches, especially for uncovering non-cell autonomous phenomenon. Now, one such example that I like to highlight was from a paper from 2019 where they showed that Parkinson's disease patient-specific astrocytes resulted in neurodegeneration of normal DA neurons. So basically, long story short, this group generated hPSC-derived astrocytes or dopaminergic neurons from either control or Parkinson's patients. And that they found that when they co-cultured the Parkinson's disease astrocytes with healthy dopaminergic neurons, it resulted in this neurodegenerative phenotype. And conversely, they saw a partial rescue when they culture the healthy astrocytes with cultured patient disease or Parkinson's disease neurons. So, and really what all of this kind of suggests is that there are clearly neural phenomena that one cannot easily or completely uncover or model using just a single cell type. So as I've mentioned, we spent a lot of time developing astrocyte and microglia co-culture protocols for many of our 2D and our 3D neural differentiation kits. A lot of those are highlighted here within that box. But this is definitely something that we're sort of building into kind of our workflow moving forward. Anytime we develop a new kit, we try to keep in mind, what other cell types are relevant if you want to actually, faithfully model using that neuron. And I do want to say that this is no small task, at least if you want to ensure long-term viability of these co-cultures. So co-cultures of different cell types can be challenging for a lot of reasons, including sort of variability with the protocol timing, cell proliferation rates, and just variable culture conditions. So, we've put a lot of effort into testing a number of different culture conditions before we could actually land on one that successfully maintained multiple cell types for extended periods of time. And just to give you a sense of what one of these hybrid protocols looks like, here's a schematic of our forebrain neuron and microglia co-culture protocol. What's not really captured within this diagram are the 25 unique steps required to really bring all of these together. So it's a very detailed an involved process that we've sort of, figured out for you. And here are some representative images and a bit of data from some buy and try cultures that were set up based upon these protocols. One thing that I just really kind of want to highlight, it's an interesting fact, which has been repeatedly shown in the literature, is this benefit conferred by co-culturing neurons with astrocytes, both in terms of neuronal viability, but also in terms of neurite outgrowth. And this is something that we also see. And to summarize everything, building a better in vitro model for neuroscience, really begins with an eye towards ensuring the protocols are physiologically relevant, easy to use, and consistent and flexible. And with regards to this physiological relevance, we feel it's important to take into considerations such as developmental fidelity, the activity and function of those cultures, as well as the relevant cell types and cytoarchitecture. And this all feeds into this idea that co-culture and 3D models are really powerful tools for modeling, not only neuroinflammation, but also other neural phenomena. And with that, I think I am just under time. I will gladly take any questions or comments or feedback.
Any questions for Jeff? So what's the best way to search on your website for the co-culture systems?
I would say you could just type in co-culture and that should come up, but there's actually a really nice section where we have basically all of our support protocols, and it should be captured under there. Or you can usually find them under all the different differentiation kits. We usually have them linked as well.
All right. Thank you,
All right.
Jeff.
Well, thank you so much.
Our